it's time to clear up some of the confusion of the electron transport chain. Now, the electron transport chain is one of the final steps of cellular respiration. It takes place after the Krebs cycle, where we have a couple products like NADH and FADH2 and 2-ATP. So it takes place after the Krebs cycle, and it helps to form an abundance of ATP, which can be used in further cellular respiration or to give energy to other cells that need it throughout the human body or whatever cell we are talking about. Now we get into it. ATP, as we should know, is a form of energy. Now, in order to form this ATP, we have to have cellular respiration, of course. We also have to have the Krebs cycle. Now, this all takes place in the mitochondria. The mitochondria is the powerhouse of the cell because it's where we form our ATP. It's where we form our energy. Now, I have a picture right here of a mitochondria, little sketch I made. And in the middle, you can see this is the matrix. Now, the matrix is just a fancy word for where Krebs cycle takes place, where cellular respiration takes place, and where the electron transport chain takes place. Now, if we zoom into the matrix, we can see that there's these things right here called protein complexes. Now, what's important about these things is they can carry an electrical charge down the gradient. So when electrons come into these protein complexes right here, or right here, it can carry it down this way toward this thing called ATP synthase. So this will be relevant in a little bit, but what I want to tell you is that the Krebs cycle is vital. Let's get into that. So the Krebs cycle forms a couple main products that we use in electron transport chain. And the reason for making these products from the Krebs cycle is for exactly that, to produce ATP from the electron transport chain. So it produces some NADH, and it makes some FADH2. We also get some ATP, but not the amount that we're going to get from the end of cellular respiration due to electron transport chain. So these molecules right here, I really want to focus on NADH and FADH2. These molecules were reduced. By the Krebs cycle. So what the Krebs cycle did is they reduced NAD positive to NADH, took away the positive charge, and did the same thing for the FADH2 as well. It was FAD, and then it became FADH2. So what occurred here is both molecules were reduced. And the reason for this is because electron transport chain, also known as oxidative phosphorylation, does just that. It oxidizes these end products from the Krebs cycle. So it's like a cycle, exactly, Krebs cycle. So we take these molecules that were reduced by the Krebs cycle, they are oxidized by the electron transport chain. Both of these are oxidized, which delivers energy, which I'll get to in a second, because there's a reason for the oxidation. It's a vital component. Then once they're oxidized, they can go back through the Krebs cycle and be reduced again back to this state. So it's a cycle of, eventually we'll get to it, it's a cycle of donating and accepting electrons. That's the whole name of the game when it comes to the electron transport chain. A little bit of a tongue twister there. So let's get into oxidation, just to make sure we have a good understanding of it, because sometimes it can be a little bit confusing if you haven't heard about it multiple times. So oxidation, we're gaining charge, we are losing electrons. So this would be occurring during the electron transport chain step when we go from NADH to NAD positive. We're gaining charge. We're gaining this charge. Now, FADH is going to do this as well. It's actually going to lose 2 hydrogens and become FAD. It loses electrons, it gains charge. So that's for oxidation. This is what happens in ETC. So this is exactly what happens. You lose electrons in electron transport chain. Now, reduction occurs, this occurs in the Krebs cycle. In the Krebs cycle, you are going from those end products of the electron transport chain, like your NAD positive and your FAD, and they are being reduced. Let's put in a different color. It's going to be reduced to NADH and FADH2. So that's for reduction. The Krebs cycle does this right here. So the Krebs cycle reduces these molecules so that the electron transport chain can oxidize these molecules. Oh, let's go back up. So, how does this work when we're speaking about the actual process that occurs? Because the process is actually relatively simple once you have an understanding of oxidation and reduction. So, let's go with orange. All right. So we have our reduced molecules, NADH. We have FADH2. I'm not going to get into like the details of each. There's different. I believe there's more NADH2 than FADH2 in the process. However, the function of each is what I really just want to stress. So we take NADH2, goes into this electron, into this protein complex. It loses electrons, so therefore it's oxidized into NAD+. Now what happens to these electrons? Well, the electrons, like I said before, will ride these protein complexes down the belt all the way down to ATP synthase. So we'll see that FADH2 does the same thing. It's going to lose its electrons and its hydrogens as well, which I'll get to in a second, and it will become FAD. Now, the thing to understand with this is that we also lose hydrogens. So these hydrogens get pumped out of the protein complexes and into the extracellular fluid. Now you're going to have a lot of these protein complexes on the membrane of the matrix. So it's safe to assume that we'd have a lot of hydrogens out here in the extracellular fluid, all carrying a positive charge, right? So we have this buildup of hydrogen outside of the cell. And if we can remember back to gradients, so when we're speaking of gradients, by passive diffusion, we move from an area of high concentration to an area of low concentration. So there's a low concentration of hydrogen inside the cell. There's a high concentration of hydrogen outside of the cell. So this means that once we get something to open, once we get a passageway to open for the hydrogens to move back into the cell, it's going to be a rush of hydrogens into the cell, back in. And you can actually take advantage of this gradient, if you will. So back to when these electrons were moving down the protein complexes, right? It triggers ATP synthase, which is just that, it synthesizes ATP to open its gradient channel. And when it does so, since the channel can open, all these hydrogens can move down through it. Now, ATP synthase is actually a very complex molecule. It acts as a motor. It spins a motor that actually generates ATP. So once this moves, all these hydrogen move through here, we actually do generate ATP, and we generate a lot of it, up to over 30 molecules of ATP